When we get a cut, the blood tries to leave our body. There are proteins inside the body called clotting factors that work together with the platelets and other cells of the body to prevent the blood from leaving. In hemophilia, it is a disease where one of these proteins doesn't work properly, and that upsets the whole structure. So in this video, we are going to cover the background of the molecular background behind why hemophilia behaves as it does. In another Molecular Moments video. This might look rather intimidating, and it's okay because you don't need to know it for this class. This is what's called the Clotting Cascade. It turns out in order to get blood to clot, there's a lot of pieces that have to come together, tons of different proteins that all interact with each other and work in what's called a cascade to activate each other in sequence in order to get the end result, almost like runners passing off a baton. And these factors have been given numbers in Roman numerals because there are so many of them. And each one requires a certain circumstance to turn it on, and then it goes and turns on another one down the line until in the end we get fibrin that forms or that interlinks blood cells in order to form a blood clot. Well, it starts out, one of the pathways starts out with factor 12, which is a protein that flows around in your blood. So this is like a little blood vessel here. And it's gotten a cut right here on the wall. Factor 12 bumps into collagen, which is underneath the blood vessel cells, and that turns it on, and it becomes 12A. Well, when it becomes 12A, it now changes shape. Remember how earlier in the semester we talked about how shape of a protein determines its function. Well, in this case, the shape of 12, once it bumps into the collagen, it changes shape and becomes an enzyme that's actually capable of turning on this other protein. So protein 11, clotting factor 11, gets turned on, activated by clotting factor 12. So that turns factor 11, it changes shape and becomes an enzyme itself. Factor 11 goes and hunts down factor 9 and uses its enzyme activity to turn factor 9 into an enzyme. Now factor 9, its job is to find factor 10 and turn factor 10 on. And factor 10 is supposed to find prothrombin and turn it into thrombin. And thrombin is supposed to go find fibrinogen and convert it into fibrin, which then gets pieced all together into a blood clot by factor 13. Over here somewhere. Here we go. Whoop. Factor 13. Oh, that's correct. Okay, here we go. So factor 13. Well, it turns out that factor 9 needs helpers. It turns out that in order for it to be able to do its job, it needs help from another little protein called factor 8. Factor 8 comes in and helps factor 9 turn on factor 10. If you don't have factor 8, then you can't turn on factor 10 and nothing downstream happens. And if you don't have factor 9, then you can't turn on factor 10. And so again, nothing downstream happens. Well, hemophilia is a mutation in what's called the factor 8 gene, or the F8 gene. And genes, by convention, are always italicized with the gene name. So it's the F8 gene. And the F8 gene, when it's transcribed and translated, which we went over earlier on in the semester, when it's transcribed and translated, it codes for a protein called factor 8, clotting factor 8. And clotting factor 8 helps clotting factor 9 turn on factor 10 to turn on prothrombin to turn on fibrinogen and cause a blood clot to happen. Well, in hemophilia, when we break the factor 8 gene, it doesn't make factor 8 it doesn't make working factor 8. So factor 8's gone. Now, if factor 8's gone, that means factor 9 can't turn on 10 because this is gone. Which means if factor 9 can't turn on 10, then 10 doesn't get activated, which means prothrombin doesn't get activated, which means thrombin or fibrinogen doesn't get activated, which means fibrin doesn't get converted into a blood clot, and all of this is gone. We don't have a blood clot. And it doesn't matter if these other guys get turned on, because 12 can activate 11, can activate 9, and all of that will work just fine. But with factor 8 broken, then factor 9 can't turn on 10, and we fail to make a blood clot, and so they can bleed out with just a small cut. So it's very dangerous. And so here's an example where one protein can really mess them up. Now, there are more than one kind of hemophilia, and each one is just a different protein involved in clotting that's a problem. So hemophilia A is the more common one, and hemophilia A is a broken factor 8 gene. Hemophilia B is factor 9 that's actually broken. And then there's another one called von Willebrand's disease, and von Willebrand's disease involves another protein that's involved in clotting that's not shown here. But all of them result in the same problem of hemophilia, in that no matter which of these pieces you break, you can't get the message down to fibrin to activate the blood clot, and so it's not going to work. So that's why hemophilia behaves the way it does.